Assalamu alaikum. Today our topic is OPERON. OPERON is also studied for the regulation of gene expression in procreation. As we know, bacteria produces a specific enzyme and proteins in order to meet its metabolic demand and also to reproduce. What this bacteria produces the raw material like the amino acids and enzymes, etc. Among these amino acids, one of the essential amino acids, tryptophan, is also produced. Now the process by which the bacteria produces the tryptophan is a OPERON. So what is a OPERON? OPERON, first of all, it is an operon system. That means multiple genes are controlled by a single promoter, and always the product of OPERON are the enzymes that convert the racemic acid into the tryptophan in a series of the steps. By using this operon, bacteria are able to produce the tryptophan, which are used in a protein synthesis. This operon was first characterized in Escherichia coli. It is the first repressible operon to be discovered. Now we will be talking about transcriptional regulation of these operon genes. This operon contains the 5 genes TRPE, TRPD, TRPC, TRPB and TRPA. TRPE and D produces the enzyme anthranolate synthase. TRPC encodes for the indole glycerol phosphate synthase enzyme. TRPB and TRPA encode for the tryptophan synthase. These all are necessary for the production of tryptophan amino acid. Like the lactopron, tryptophan also has depressor region, and the regulatory region includes promoter region, operator region, and in addition is the leader region or the leader RNA sequencer and the structure genes. Now assume the situation when the tryptophan level is high. When the tryptophan level is high, it has been seen at that situation operon status is off, and when the tryptophan level is low, then the operon status is on. That means the operon genes transcribe and produce the tryptophan amino acid. It makes sense when the plenty of the tryptophan, then bacteria not waste the energy, and when the tryptophan is low, then produces it. So we can say that it is an on-demand process. By the default, tryptophan operon is on when the no tryptophan is present in the bacteria. Now just look at what happens when the tryptophan level is low. When the tryptophan level is low, then operon genes all are transcribed and produce the enzymes that are necessary for the synthesis of the tryptophan amino acid. How does that happen? So like the operon, there is also a repressor, and this repressor is known as a aporepressor. This repressor, in the absence of the tryptophan, cannot suppress the transcription of the genes. When the tryptophan is high, then this repressor is known as a hollow repressor, and this repressor suppresses the transcription of the genes. RNA polymerase cannot bind and not transcribe the genes. Now if we zoom in this situation, promoter and operator system also have a leader sequence, and this leader RNA has 4 specific sites, 1, 2, 3 and 4. 4 specific sites which can pair with each other. 2 and 3 pair and known as an anti-attenuating stem loop. 1 and 2 can also pair, 3 and 4 can pair each other and forming an attenuating stem loop, which results pausing of RNA polymerase activity and not transcribe the genes of operon. Now we look at the situation when the tryptophan is high, then 1 and 2, 3 and 4 can pair each other and forming attenuated stem loop. That is also known as a terminating stem loops. Here is the highlighted sequence shows as in blue in color. It means that this region has uracil rich region. That means the greater chance of RNA polymerase pausing activity and genes not transcribed in this situation. When the tryptophan level is low, then anti-attenuating stem loop or the anti-terminating stem loop 2 and 3 can pair, forming anti-attenuating stem loop. And all the tryptophan genes are transcribed. Zooming, here is the 1 and 2 when paired, when the tryptophan level is low, then forming anti-attenuating or the anti-terminating stem loop. When tryptophan is high, then 1 and 2 can pair with each other, 3 and 4 can pair with each other and forming the terminating or the attenuating stem loop and pausing the activity of RNA polymerase. Zooming in detail, bacterial transcription and translation are coupled together. Imagine a situation when the tryptophan is high, ribosome speed is also high to form a protein. Then 3 and 4 can pair with each other. 3 and 4 and 1 and 2 loops are the attenuated loops that terminate the RNA polymerase activity. When the tryptophan is low, there is a specific site code for the codon corresponding to the tryptophan. But you can understand when the tryptophan level is low, as a result, amino acid transfer RNA which brings the amino acid or the tryptophan is also less in abundance. So as a result, ribosome speed is also low and 2 and 3 can pair with each other and form an anti-attenuating stem loop or the anti-terminating stem loop. RNA polymerase move forward and transcribe the genes and give rise to the protein which necessary for the conversion of the tryptophan. When the tryptophan level is low, then ribosome speed is also the low and the transfer RNA that are necessary for the transmission of the tryptophan amino acid into the ribosome surface area. Then 2 and 3 form a pair and this cannot stop the movement of the ribosome or the RNA polymerase. RNA polymerase move forward and the transcribe the genes and give rise to the protein which is necessary for the conversion of racemic acid into the tryptophan amino acid. So tryptophan is an anabolic pathway which requires lots of the energy. That's why bacteria not waste the energy when the tryptophan is present in the bacteria. When tryptophan is absent, then the tryptophan is on and all the genes are transcribed to produce the tryptophan amino acid. Gene regulation is more complex in case of the eukaryote than in case of the prokaryote. This is all about the tryptophan. Thank you so much for watching.